Hello, I'm Alice Popejoy, and the title of my talk today is Population Diversity and Polygenic Risk Prediction, Implications for Implementation in Genomic Medicine. I have no financial disclosures. Now, in all areas of science, the amount of data that we have, the type of data that it is, and how we conceptualize that information is going to limit our ability to make accurate predictions, and we're going to make errors in terms of how we classify what we see. One of my favorite examples of this is when a group of anthropologists in the late 1800s discovered a Viking warrior grave site and determined it to be a male individual, despite the fact that the bones more resembled the morphological features of female bones at the time, due to gender stereotypes. And in 2017, a group of Swedish geneticists discovered that, in fact, this was a female individual by doing genetic analysis on those remains, and they did not find a Y chromosome in the sample. So this is just an example of how our cultural frame, the time that we're in, and how we conceptualize what we see or filter it through our lenses is going to affect how we conduct science in medicine. So in the United States, there's a long history of racial classification that is based on underlying ideologies of scientific or biological racism. This goes back to Linnaean taxonomy in the 1700s, where Linnaeus categorized humans into different groups as if they were different species based on race or very rough physical appearance and geographic location. In this image here on the left, you see Types of Mankind in 1854, where there was considered to be a hierarchy of races, with the sort of Greco-Roman skull being the pinnacle of humans, and having a gradation of humanity down to chimpanzees. And this idea that there are fundamental biological differences between humans on the basis of race pervades science and medicine. These images here are from the Jim Crow era, where there were segregated hospitals and clinics, and all of that is based on the belief that there is some fundamental difference between races of humans that necessitate differences in medical treatment, which we know is not the case. And unfortunately, this legacy lives with us today in our models for our biomedical research and in clinical algorithms, and this has become a very important topic of research for many people in the last. So we've known since the 1970s that these ideologies are not based in science. Of course, we know that there is more genetic variation within these racial categories than between them. And in the 1970s, we had population geneticist Dick Lewontin argue that racial classification is of virtually no genetic or taxonomic significance, and so no justification can be offered for its continuance. And other population geneticists have subsequently validated his findings and made similar inferences. However, despite the fact that we've known this lack of genetic differences that differentiate people by race and ethnicity, we have a system of racial and ethnic classification in the United States that is based on the categories for the census that is used ubiquitously throughout biomedical research. Now, part of this is because in 1993, the NIH was directed by Congress to require reporting of demographics based on the OMB census categories for all of their funded research projects. And an unfortunate consequence of that is that researchers tended to interpret these categories then as the sanctioned analytic categories that they could use in their research protocols and analyses. And indeed, it's been integrated into our electronic health record systems, and we see on clinical genetic lab testing forms various combinations, expansions, or reductions even of these categories that is really heterogeneous across labs and healthcare systems, but indeed is used in almost all of them in the United States. But there is not a lot of conceptual clarity around what these categories actually mean and how we think about them. So whether Asian, for example, is a race, an ethnicity, or an ancestry really depends on the context and how the person is using it. And if you're thinking about Asian as an ancestry category, well, that's roughly half the world's population. So what does that really tell us about someone's underlying genetics? Not much. In the UK, there are similar categories, also based on the population census, and I've tried to sort of contextualize these in terms of whether something is a race, an ethnicity, or an ancestry, and you can see there's a lot of fluidity and overlap. I also added nationality here because that's an important political category in the context of the United Kingdom. And these are the population categories that are used in the UK Biobank. So you'll see here on the left how the breakdown occurs across these different categories, and I would encourage folks to consider why they're listed in the order in which they are. So there are no universal definitions of race, ethnicity, and ancestry. It's a topic of inquiry for anthropologists and social scientists, and that's not my area, but I did lead a group in 2019 of international interdisciplinary researchers as part of the Psychiatric Genetics Consortium special interest group in genetics and in population genetics as well. And it was difficult because it was an international group, so there were lots of different opinions, but ultimately we landed on these definitions, and you can see them in the paper. And what I think is important to note is that race is almost always linked to social power dynamics. And certainly racism. You cannot separate race from racism, and that's why we can see inequities that are affected by systemic racism that do have an impact on environmental risk for complex diseases and can also potentially interact with genetics to affect a person's risk. Ethnicity is a different term, but it is often used interchangeably or just with a slash with race. There's not a universal agreement on how ethnic groupings should be characterized. Some ethnic groups may share genetic factors due to having similar ancestries, whereas others can be strictly social and cultural in nature and not have much to do with genetics at all. And finally, ancestry varies a lot by context. In the case of genetics, we're talking about genetic ancestry, whereby we estimate somebody's ancestry proportions based on how much of their genotype information matches up with that of reference populations. So this is, again, limited by the information that we have from existing population data sets. And this sort of labeling of people's ancestry, which we can have many, many ancestries across all over the genome, depending on chunks of chromosomes that are inherited through different lineages, maternal and paternal. But we really oversimplify the complexity of human genetic variation when we do this. But that's not to say that there aren't important differences by genetic ancestry. There are systematic differences in allele frequencies and in patterns of linkage disequilibrium across ancestral populations. So we can look at people's genotype information in different geographic regions, as they've done here in this paper on the left, looking at African ancestry clusters, that is different parts of Africa where there are reference populations, and then looking at those proportions of African ancestry in the Americas. And so you can see that there are different sort of geographic clusters of ancestry groupings. And so this is a real phenomenon, and there are declines in allele frequencies across regions. So it's not to say that there aren't differences, but the majority of genomic variation that is found in human populations worldwide is shared among all different groups. It's just that the frequencies of those differences vary across geographic regions. So I really like this image here on the right from a paper in Science Education, helping us to understand that the majority of genetic variation is shared among groups. I think that's an important point.